Okay, good morning. Let's start the class. We already finished the 3 lab sections, so let's do some of the review of what we did before, especially go over some of the results. Okay, so first of all, we did the simple stain. And the simple stain, you're seeing 2 bacteria, which is E. coli and Bacillus subtilis. Now, what are the simple stain results tells us? Morphology and arrangement. E. coli is small, short, single rods. Bacillus subtilis, we call it a large, single rods. So both of them are rods. Okay, when you say the bacillus looks like it's a lot. We did the gram stain. And the gram stain, we're using the 2 bacteria, which is the E. coli and Staphylococcus aureus. And I said the gram stain, we have to record 3 important information. So what we find, Staphylococcus aureus is gram-positive, coxile, and grape shape. You can say grape cluster. E. coli, it is pink after you do the gram stain. So it is gram negative, it's rods, and we can say it is single short rods. So these are the things you should record in your notebook, and also better have a picture to show what you did. And I believe in the lab menu, if you look at the color PDF version in the eCampus, you can see the results there, and I put the results there already. Okay, so this is what we did already. And today we will move on to the lab 4, another major stain, which is called Endospore stain. So first of all, we would love to briefly mention what is Endospore. First of all, there is 2 major bacteria has Endospore, is Clostridium and Bacillus. We will not do the Clostridium here in the lab. The reason is Clostridium. We talk about Clostridium perfringens, Clostridium tetanus, Clostridium difficile, and the Clostridium botulinum. Those are class 3 biosafety level, BSL3. This lab is only certified for BSL 1 and 2. So we cannot do that. We are allowed to use Bacillus. Today we are using is Bacillus cereus. And also we know Bacillus have another species which is subtilis. So this is the first thing you need to know. Second, what is Endospore? Endospore is a protection structure, which means is in a very stress environment, they will be forming endospore, like UV light, very cold, acid, desiccation, all those stress environments. Now, what are the endospore looks like? Same thing as what we talked about before. There's a multiple layer. And the outside is called cortex. Then you have a membrane system. And the cortex, the major component is polysaccharides, same as we talk about the peptidoglycan. Now, of course, outside you have another layer, which is talk about is we call it a coat. But why endospore could be survive in a stress environment? The key is right in the center. There is a two very special structure will let endospore survive and protected in stress environments. Number one is calcium diproconic acids, DPA. Calcium diproconic acids. Okay. Diproconic acid. Another one, there is another special structure called small acid soluble protein. And there is a third question. Why we want to do the endospore? Because we want to see the presence, absence of endospore. This is the first thing first. The second, we need to know the location of the endospore. Because the location endospore is genetic dependent. And we mentioned this in the lecture. It could be the endospore is right in the middle. We call it a central. It could be at the end. We call it a terminal. And most of the time is right in the middle. Between center and the terminal, we call it a subterminal. And the location is genetic dependent. Bacillus most of the time is subterminal, Clostridium some of them are terminal and the center. So we'll see. Okay. Now, how are we gonna do the endospore stain? Then we talk about the stain procedure. Majority of the steps are the same. So, number one, you're gonna get a glass slides. Draw a big circle in the middle. That's called the smear preparation, the same thing. This time we use Bacillus cereus, so we write BC. And then you add one drop of the, flip it over, okay? Add one drop of water. And then make bacterial smear. So you add one drop of the water, you use loop to pick the bacteria, and then you mix it very well. Okay. Then what you do? Air dry, same thing, 5 minutes, and then what we followed by heat fixing. These are the same. Now today, once you finish right here, we're gonna have two groups to do the next major step. Because endospore is, we call it a rigid, very tough structure. So we're using a basic dye and not gonna come in. We need to use a special dye. This special dye is malachite green. It is very green. And we need another major step is called heat steaming. And how to do that? When you have this smear preparation already, okay, let's say everything is ready. You had a bacteria there, the smear is there. That is Bacillus cereus. First of all, we need to flooding with malachite green. This is one of the group you're going to do is flooding with malachite green. What means flooding? You need to cover the whole glass-like surface with malachite green. And then I will use Bunsen burner right here. A Bunsen burner to do back and forth steaming. for 5 minutes. This is one of the methods. We will have 2 groups and working with me, I'll do it. Then have another group we're working with here, how we do it. That's a little bit easy. Same thing, there's a glass slides right here. Okay, you make a smear. We will using water bus to heat it. So you only need to cover the malachite green is right in the middle. So this will be adding malachite green. And then we're going to put into a water bus. You see the water bus is already turned on. And the steam is coming from the bottom. And this will be steam about, let's say we do 2 minutes. Sometimes we do 1 minute. Let's do 2 minutes. Let's steam a little bit strong, okay? Then hopefully it will be penetrated. Now after that, very important step, go ahead to cool down. You have your gloves and your faucets is ready. Don't use bare hand. It's very, very hot. Don't burn yourself. Okay, have the gloves and have the faucets cool down on the rack of the water tray, and then we gonna to rinse it. So gentle rinse. And then we counterstain, we add the saffron. Okay, we add the saffron. Now, what gonna be the Endospore looks like? When I tell you, majority of them will be seen not like your picture. Not the picture like an artist in your textbook, no. It will be very similar. You're gonna see it's a bunch of green. Okay, they are all green here because of malachite green. You will see there is a big bubble-ish right in this middle. Right here. This is kind of thing like Endospore. Now the last thing I want to mention, it is different from gram stain. Endospore stain, we need to use old culture because we need to let the bacteria to have Endospore. So usually 35 degrees Celsius, at least 72 hours, will let the Endospore forming. Bacillus cereus, the strain we use in the lab, is a little bit different. It is growing Endospore a little bit fast. So we did like 48 hours. But keep in mind, Endospore stain, you have to use the old culture because we want Endospore to forming, and the gram stem, you have to use a young culture because it's rely on the principle is cell wall structure, is peptidoglycan. So there is something difference, okay? Now, I'm not going to show you again about everything here because you already know how to do. Everybody, go ahead into the front desk to get Bacillus subtilis right here. So then it's BS, Bacillus subtilis. Make a smear, air dry and heat fixing. Then, these 2 groups have all your glass slides put on the rack, okay? Those 2 groups have all your glass slides, work with Ryan, put on the water bus, and we are going to do the heat steam. But both of them are very hot after that, so be careful, wear gloves, use faucets, not bare hand. Okay? Then we can go ahead to do it. And you will have fun. Trust me, that the steaming is very interesting to do.